In an earlier lesson, we learned what it means to say that A causes B. That's important because causation is a key concept in science. Science is really an attempt to explain what's happening in the world, and to explain something is to give the cause of something, so we better know what we mean when we say A causes B. And when scientists make a first attempt to explain something, it's called a hypothesis. So a hypothesis often says something like A causes B. Now, the method of science is designed to test the truth of these hypotheses. So an experiment is designed to test the truth of the hypothesis. So we can think of science overall, in general, as hypothesis testing. Experimental design is based on the definition of A causes B. And we learned in the previous lesson that when you block A to see if B happens, that was the control condition, and when you supply A to see if B happens, that was the experimental condition, where A would be the variable. We'll use our scientific understanding and apply it to the case of frog deformities. In the mid-1900s, first some students on a field trip noticed a pond where there were frogs with high levels of deformities, and then as scientists and ecologists became interested, they began to discover high levels of frog deformities in ponds all over North America. Now, what the students found were frogs like this. They had missing legs, sometimes the tail wasn't growing right, and sometimes they had extra legs. So we'll call this the case of the frog deformities. It raised an obvious question: what was causing the frog deformities? Just a reminder, frogs begin as tadpoles in the water, so they swim around, they have gills, and they eat stuff in the pond or the stream, and then they go through metamorphosis, where they grow limbs, and then the tail will disappear, and then this four-legged animal will be able to hop up out of the water onto the land to feed and such. So, an early hypothesis was that maybe pesticides were causing deformities. The pesticides maybe used on nearby fields would run into the ponds, and then, the high levels of pesticides might be influencing development and causing the deformities. A reasonable hypothesis. So scientists did what scientists do. They design an experiment where you have two groups, one group of tadpoles, they don't get any pesticide, the other group of tadpoles gets pesticides. What they found was that high levels of pesticides did in fact kill tadpoles, but they didn't cause deformities. So the result does not support the pesticide hypothesis, because the pesticides did not cause the outcome that we were trying to explain in the first place. So the pesticides are toxic in high levels, but they weren't causing the outcome that we're trying to explain. So the pesticide hypothesis was weakened by those early experiments. Another hypothesis presented itself when scientists began to examine closely the deformed frogs. What they found, you can see in these pictures here, was right at the place where there was some kind of deformity, like a missing leg here. They found parasites, living organisms that were living at the site of the deformity. They're also present here, harder to see. So it raised the possibility that maybe as tadpoles, the parasites somehow got into the tadpole at just the places where perhaps legs were later going to form, and interfered with the formation of legs. So in this case, maybe the leg would not grow, or the parasites, just by the nature of where they inserted themselves, could influence development in another way to make extra legs. So this we'll call the parasite hypothesis. Now, one of the early observations made was in Northern California, where there was a region that had a whole bunch of different ponds. Now, remember, the pesticide hypothesis was still sort of on the table early on, so we've got two hypotheses, the pesticide and the parasite hypothesis. Now, what the scientists wanted to do was simply make some observations about frogs and parasites and pesticides in a set of ponds up in Northern California. And here we see the results of that observation. So each of these would be a little pond, it would be separated, and where they found parasites in the pond, they found frogs with high levels of deformity. So in this pond had high parasites, high deformity, parasites, high deformity, parasites, high deformity. In ponds without any parasites, there might be frogs, but they didn't have the deformities. And in none of these particular ponds did they find any significant levels of pesticides. Now, this kind of observation is a correlation study. And it's an important type of study that scientists make use of, but as we'll see, there are limitations to this type of study. A correlation between event A and event B just means that there's a regular relationship between A and B. So in ponds with high levels of parasites, there were frogs with deformities. In ponds with no parasites, there were frogs with no deformities. So the fact that you see parasites and deformed frogs in the same ponds, and you don't see the two occurring together in the other ponds, that's evidence of a correlation. That's a regular relationship between parasites and deformities. Now, you might be tempted to say that this is evidence that parasites are causing deformities, but we have to be careful here. A correlation between two things does not, by itself, establish that one is causing the other. In fact, there are 3 ways to explain a correlation. In fact, there's more than 3 ways, but we're going to look at 3 important ways to explain a correlation. If A and B are correlated, it could be that A causes B, so we'll say A arrow B. It could be that B is causing A, B arrow A. Or it could be C, a third factor is causing both A and B. Notice the way we're going to write that. A third factor is causing both A and B. And again, the fact that A and B are correlated just kind of means that there's a regular relationship where you find A, you often find B. Well, that would happen if A is causing B, or B is causing A, or C is causing both A and B separately. So, to our frog case then, the fact that we find a correlation between parasites and deformed frogs, it could be that the parasites are causing deformities. Now, this would be of interest to ecologists, and it might be the thing that leapt to your mind, but we have to consider 2 other possibilities. It could be that deformed frogs are somehow causing high parasite levels in those ponds where the deformed frogs are living from some unknown mechanism at this point. Or it could be a third factor is causing both parasites and deformed frogs to show up in the same ponds. So it's a very important lesson in science that correlation is not the same as causation. It might be suggestive. It's very interesting to try to establish correlations between things in the world, because sometimes one thing is causing the other. But to establish causation, we need to do an experiment. All right, so it's time to test the parasite hypothesis with an experiment. We'll define the variable then as parasite exposure. So the tadpoles will be exposed to parasites, so the parasite exposure will be our variable. That's the thing of interest. That's the thing we think is causing the effect. We think parasites are causing deformities. The control condition then, there's going to be no parasite exposure. And the experimental condition, we'll set up 3 exposure groups, a low, medium, and high. So we're going to control the number of parasites in each of these 3 groups here. These will be 3 experimental conditions. So to test the hypothesis, notice what we're doing. We're blocking A to see if B happens, and that would be the control condition, no parasite exposure. We're going to supply A to see if B happens. That's going to be the experimental condition. In this case, we're going to get a little more sophisticated. We're going to have 3 different levels of parasite exposure. And then we're going to compare the results, and notice what we're doing in an experiment is we're gaining control over the variable. All right, this graph shows the result of the experiment. So on the y-axis we have percent deformities. So maybe each condition had 100 tadpoles, and so we're just looking at what percentage of the tadpoles ended up showing deformities when they developed into frogs. On the x-axis we have our conditions. We have the control condition and 3 experimental conditions. Now if you just take a look comparing the control to the experimental conditions, the fact that we get deformities in all 3 of the experimental conditions and no deformities in the control condition, that is strong evidence that parasites are causing the deformities. But beyond that, notice what happens as you go from low to medium to high. In the parasite exposure, you get an increase in the number of deformities. This is called a dose-response effect. We'll just call it the dose effect. By increasing the dose, right, the amount of parasite exposure, you're getting a greater effect. That also is extra evidence that parasites do cause deformities. So when you do an experiment like this, when you gain control over the variable, you can make conclusions about causation. When you're just making observations and looking to see whether there are regular relationships between two interesting things like parasites and deformities, that's a correlation study. You cannot make conclusions about causation here. You can only conclude that there's a correlation between parasites and deformities. You do not have control over the variables when you're just making an observation like this. To make conclusions about causation, you have to do experiments. You have to gain control over the variable and then compare the outcome to the control condition. So, in summary then, our definition of A causes B, A before B, block A, no B, supply A, B. And notice the definition of A causes B helps us design an experiment. So the block A, block the variable, that's going to be the control condition. No parasites. Here are the frog eggs turning into tadpoles. And then we had a couple of experimental conditions. Maybe you add a few parasites to this condition. You have a high number of parasites over here. And the result is shown down here in our simplified bar graph, that as you increase the number of parasites, you increase the number of frogs showing deformities. The control condition had no deformities. This is powerful evidence that parasites are causing deformities, and the dose effect is even more evidence that parasites are causing frog deformities.